of anchoring junctions and tight junctions. So I think we finished class today with this slide here looking at anchoring junctions. These are junctions that allow cells to be tightly bound to one another so that movement of a large organism is not going to dislodge any of those cells. You see that there are linker proteins essentially that either link two cells to each other directly by way of their cytoskeletons, or they link a cell with the extracellular matrix. If you've forgotten what the ECM is all about, go back to chapter 4 and read that section on the extracellular matrix. It's a topic that's going to come up often in this class and then later in advanced cell biology if you go on to take that class. So learn the terminology: adherins, junctions, desmosomes, hemidesmosomes, and focal adhesions. And learn the different types of proteins that are involved. The cadherins, which are both involved in attaching cells to one another by way of their cytoskeleton, either actin filaments or intermediate filaments. And integrins, which attach cells to the extracellular matrix either by their intermediate filaments or by actin filaments. So make sure you study through that, and if you have any questions, let me know. Here are some images of the different types of proteins, the cadherins and the integrins. So if you take a look at these, cadherins are involved in linking cells to each other. So this white space here that you see, I'm down in letter A, this white space here is the gap between the cells. And the cadherins are the proteins that are overlapping with each other that are crossing the membrane and then by way of a linker protein attaching to the actin filaments within the cells, or in some cases to the intermediate filaments, depending on which type of junctions we're talking about. Over here we've got integrins that you can see integrin membrane spanning here over on the right hand side, linking to the two collagen fibers by way of a protein called fibronectin. Collagen and fibronectin are extremely important molecules in disease, so they're worth getting familiar with. And then on the cytosolic side, we've got linker proteins attaching to, on the cytosolic side, I said that right. Linker proteins attaching to actin. So I think what I said backwards, was that collagen fiber and fibronectin are very important extracellular matrix polymers. So spend some time getting familiar with the terms and the basic definitions, spatial locations, and functions of these different types of molecules. Last but not least, we have tight junctions. Tight junctions are almost always seen along with the anchoring junctions that we saw 2 slides back. Tight junctions help to seal a layer of cells. So think about a place where you might want to see tight junctions. A classic example is in the intestinal tract. The epithelial layer of cells lining the intestines is only 1 cell layer thick, and immediately on the other side of it you have blood vessels. You can't have pathogens or toxins leaking through and getting into the bloodstream. So tight junctions are used to essentially seal off and almost make them, you could think of it as being waterproof, though that's not exactly the purpose, so that liquids, pathogens, other materials cannot cross between those cells. So get a look at tight junctions. You can see it's a series of proteins called occludin and claudin that are involved in creating these tight junctions. You've got the plasma membranes of the adjacent cells, and they're going to be pinned together, and you can see up in this upper right image, it's almost a stitching, and it wraps around like in a band type pattern all the way around, as opposed to the other junctions that were more like rivets, or a snap or a button. An individual junction at a single spot on the cell, as opposed to this is more like, a belt going all the way around and cinching them tight together. So spend some time going through these different types of junctions. Make sure you understand all the terminology. I think this would be a great opportunity or a great example of a time when you want to use flashcards. Memorize the terms, and then once you got terminology and basic definitions memorized, then go back and take a look at how they work and make sure you actually understand them beyond simply the definitions. So that's where we're at, and I hope that was helpful to you guys.